Okay, so today we are going to continue with our discussion of the respiratory system before we move on to the kidneys and the renal system. But just recap of your exam, a little rougher than the last exam. I will go through that and see if there are any questions that were consistently misunderstood. But this is where we're at for right now. Okay. So we're going to get squared away, just kind of powering through this material so we can all get going, get ready for our break. I think we can all use a rest. I think we're, at least from that, I think we're kind of dragging. Okay, so this is a reminder of some of the things that we've seen that are related to this next topic. Okay, so we're going to be talking about oxygen transport in the blood today and a bit about carbon dioxide, but the oxygen is going to be the major topic. So we know oxygen is coming into the lungs when you breathe air in. It's moving through those arteries down to tissue. It's being released at the tissue, and then it's returning to the heart through the veins. Okay, so we have a lot of numbers that we could associate with this. So over here, we have the partial pressures that we spoke about, I believe, last week, showing our partial pressures of oxygen and carbon dioxide at 100 in the alveoli, coming to equilibrium as we enter the arteries. So our pulmonary veins going back to the heart, and then the systemic arteries pumping it out, dropping to 40 at our tissue, and then staying at 40 until we return back to the heart and the lungs. Okay. So that's what we see going on as we go through our circulatory system if we're paying attention to the oxygen. So this other image on the right is from way at the beginning of our respiratory unit. So what this is showing you is just amounts of oxygen and carbon dioxide. So in milliliters per minute that we're pumping around, as opposed to in partial pressures. So what we're seeing here, right, is the exchange of oxygen and carbon dioxide at the tissue as well, about 250 milliliters per minute. We're basically replacing that with the air we are breathing in. So we see higher concentrations of oxygen in the pulmonary veins and really the systemic arteries. We see lower concentrations, but actually nowhere near zero when we're looking at veins. So when we say deoxygenated blood, we're not saying all the oxygen is gone. We're just saying that the level is lower. Okay. So that's the kind of basis for what we're talking about today. The other thing we will be touching back on is from our previous unit when we were talking about the blood. So we're going to be talking about how oxygen is carried. So we want to remember hemoglobin. So this is from our blood unit when we were talking about the erythrocytes, those red blood cells. We took a look at hemoglobin. So we know that hemoglobin is made up of 4 little subunits. So those are the globin, they're in red, right? So we have the globin here, globin here, globin here, globin here. So 4 of those. And then in the center of the globin parts, we have those heme groups. Again, 1, 2, 3, 4, these little flower things, which contain iron in the center. So that's what's going to be grabbing onto our oxygen as we talk through this today. Okay. So we're beginning with the transport of gases in the blood. This is where we are. Okay. So we want to be tracking our 2 main gases that we need. We have our oxygen that you need to feed respiring tissue, because you need that oxygen to break down glucose to make ATP. And then we also need to carry carbon dioxide in the blood, because that's our waste product. That's what we pull off of the glucose when we break it down. So we want to get rid of that. Okay. So we're first going to talk about the oxygen part and then the carbon dioxide part. So oxygen is going to be moving around your body, in your plasma, in your blood, in the erythrocytes. Most of your oxygen is going to be bound to that molecule hemoglobin. So hemoglobin is really mainly for carrying oxygen, although some other things can bind to it. This is important because we mentioned oxygen and carbon dioxide solubility in our last unit. And we saw that oxygen isn't actually all that soluble in water, and therefore it's not that soluble in plasma either. Right? Carbon dioxide had a much higher solubility. You could get a lot more carbon dioxide into that liquid versus oxygen. Okay, so oxygen needs to be transported by hemoglobin, partially because of that problem with solubility. Actually, only 1.5% or so of the oxygen that is carried in your blood is just in the plasma floating around. Almost all of it is transported by hemoglobin bound into the hemoglobin. Okay, so the way we refer to hemoglobin that has oxygen versus doesn't have oxygen, we have those 4 heme groups where we can bind 4 oxygens, right? And it's not an all or nothing thing, right? You can have 0 oxygens bound in, you can have 1, 2, 3, or 4. Okay. So what we're going to do as we're referring to this, if we have hemoglobin that doesn't have any oxygen bound to it, that's deoxy hemoglobin. So removed the oxygen. If we have hemoglobin bound with some O2, we're going to be referring to that as oxy hemoglobin. So all that's telling you that you have hemoglobin with some oxygen bound in. Okay. Now, we can convert deoxyhemoglobin to oxyhemoglobin by adding oxygen. So that's what this equation shows us. So we have our hemoglobin with no oxygen bound to it, that deoxyhemoglobin. We add some O2 and end up with our oxyhemoglobin. But this is reversible. So we can also take off that O2, which we want to do when we get to the tissue, right? We want to give the tissue oxygen so that it can make ATP. So this equation can go to the right or it can go to the left. So you can put on oxygen or you can peel off the oxygen. And like other equations that we've seen in this class, this is going to follow the law of mass action. So wherever you have more of a thing, it drives the equation the opposite direction. So when we have more oxygen coming in, we're going to end up producing more oxyhemoglobin. Okay. So as we go through today, we're going to be talking a fair bit about curves, specifically something called our hemoglobin oxygen curve, right? We're going to be looking at it. Dissociation curve, actually. Missed a word. Why I'm saying that to you is because this is going to be a relationship between our partial pressures of oxygen, so essentially the amount of oxygen, okay, and how much oxyhemoglobin we have. So what our saturation for that is. So like what percent of the sites available for oxygen are actually occupied by oxygen. Okay. So when we look at that curve, it's really being driven by some of these processes, right? Like as we change our oxygen levels, if we increase it, we're going to end up with more oxygen bound to hemoglobin. Okay. So when I say saturation of hemoglobin, all I'm talking about is we have those 4 sites for oxygen binding in the hemoglobin. And if we have all 4 filled, can't attach anymore, it's saturated, it's fully saturated. There's no more room for oxygen to bind, okay? So fully saturated hemoglobin would have 4 oxygen molecules bound to it, okay? And this is just following the law of mass action, which is just saying if you have more of a thing, drives the reaction to the opposite side, okay? So here we see an image of this binding happening, okay? So here we have our hemoglobin. It's a different color, but it's that same molecule, okay? We can see as we load oxygen in the lungs, we end up with oxyhemoglobin as we have oxygen binding into those heme groups. And the opposite thing happens at the tissue. So as we're losing oxygen, what we're really saying is that we're taking off these oxygen, sending it into the other cells, taking us back to our deoxyhemoglobin. Okay. So we're going to be referring to loading and unloading of oxygen. So loading is just putting oxygen onto the molecule. unloading is taking the oxygen off. Okay. We're going to want to do both those things depending where we are. We're going to want to load oxygen in the lungs. We are going to want to unload it in the tissue. So here we see a different schematic showing us the hemoglobin and the oxygen in the blood. So imagine this is like a petri dish or a swab of blood or something, and each of these little squares or grids is a hemoglobin molecule. Okay? So these hemoglobin molecules then have oxygens bound in some different amount. Oh, they want us to pretend this is a red blood cell. That's fine. We're still looking at hemoglobin and how many oxygens are in there, okay? So most of the oxygen is not going to be in plasma, not going to be floating around in the fluid. It's going to be bound into the hemoglobin. So we can see here in this first slide that it's giving us an example of arterial blood. And we can see that most of those hemoglobin are full. Most of them are saturated. We have almost all those spaces for oxygen full up. We're just missing this one. Okay. In venous blood, which has a lower partial pressure of oxygen, okay, we're going to see that we have a different sort of state. So we've unloaded oxygen to the tissue. So we've taken those oxygens off of their heme groups, passed them into different cells. So now we have a lot more missing spots. Okay. So this is what we're going to see in venous blood. We're going to have some deoxygenated blood. So these hemoglobin are not going to be fully empty, but they're going to have a lot more slots to put more oxygen when we return to the lung. Okay. So when we talk about the oxygen carrying capacity of blood, what we're talking about is basically like how much oxygen can we fit into the hemoglobin molecules that you have in your body circulating around. So to figure that out, we also have to make an assumption about how much we're filling up each hemoglobin molecule. So if we are saying that we're going to have every hemoglobin molecule totally fill up on oxygen, all 4 slots occupied. So if it's 100% saturated, a gram of hemoglobin is going to carry 1.34 milliliters of oxygen. And then the normal hemoglobin levels circulating around in your blood are about 2 to 17 grams per deciliter. And I believe you may have done some calculations on things like this in phys lab. We're not going to be doing a ton of calculating here. We're just going to be focusing on understanding curves and sort of understanding figures with this stuff. But takeaway, if you do out the math, the overall oxygen carrying capacity of hemoglobin in the blood is that about a liter of blood can carry about 200 milliliters of oxygen. So you can carry 200 milliliters of oxygen in a liter of your blood. That's when it's saturated. So that's like the maximum. In our arterial blood, we're almost at saturation. So we're going to be 98.5% saturated. Venous blood, we're at about 75% saturation. So here's the curve I was talking about. We're going to be analyzing a bit. Okay. So what this is showing us, this isn't a time scale. This is showing us a relationship between the partial pressure of oxygen and how saturated our hemoglobin is. So this is the percent saturation. So this is our hemoglobin oxygen dissociation curve. Dissociation refers to like dissociating, like pulling apart. So this curve can tell us how much oxygen we're going to be peeling off those hemoglobin molecules in order to give them to our tissue. Okay, so not a time scale. So we want to mark down the numbers we care about really first for this curve. Right. So the reason the dotted lines are at 40 and are at 100 is that at 100, that's our partial pressure of oxygen, typically in our systemic arteries. So blood going to tissue is usually at 100 millimeters of mercury for our partial pressure of oxygen. And the blood returning to our heart is usually at 40. So that's why we've picked these 2 points because these 2 points are what we're looking at in our actual bodies. Okay. Any point, though, on this chart, right, any partial pressure of oxygen, less, more, anywhere, would correspond to a specific saturation of oxygen and hemoglobin. Okay. So the two numbers from the previous slide, that 75% and 98.5%, are on this curve. So we can see here that the arteries are at that 98.5%. The systemic veins are at 75. Okay. But if you had a different amount of oxygen in your blood, you would still have some saturation of hemoglobin. Okay. So looking at these two numbers and looking at this curve, right, in order to get from arterial blood with this 98.5 saturation to venous blood with 75% saturation, what this really means is you lost oxygen, right? So you can just compare those two numbers. So we can see here that the difference between those 2 points is how much of the oxygen you lost to the tissues, or how much of it you gave to the tissue, essentially. So the difference between those two numbers is the oxygen unloading to the tissue. Okay. So, bless you. That is the normal state of affairs, but depending what's going on in your body, we can see changes to this curve, right? With proteins, which hemoglobin is a protein, when we have other things in our blood or have other things interacting with it, they can change shape, which changes their function a bit. Okay. So other factors can influence whether hemoglobin has an affinity for oxygen, right? So a greater affinity means it likes oxygen more, or whether it has a lesser affinity, which means it's going to like oxygen less. So if it has a higher affinity, it's going to hold on to oxygen, it's going to grab it tight. If it has a lower affinity, it means it's going to let go of that oxygen easily. Okay. So we're going to see this vary in different conditions. If you're in different places, sometimes one of the factors that we'll look at is altitude. Altitude can change the affinity of hemoglobin for oxygen, but it can also change within your body, partially because temperature has an effect, and different parts of your body actually have different temperatures. Okay, so we're going to have effects on that curve, and I just pulled up that normal curve and plopped it on every slide so that we can look at how that's changing while we're looking at the words. So if you want to just sketch that out, that's where that's from. Okay, so when we're talking about the effects on this dissociation curve, it's going to be described as a change to the affinity of hemoglobin and oxygen. Okay? And what that's going to tell us is something about how easily we're letting go of oxygen and how easily we're adding oxygen on. Okay, so first one is our Bohr effect. So our Bohr effect is talking about the effect of pH on oxygen unloading. Okay, so in active tissue, it is true that you're going to be producing more acid as that tissue is metabolically active. Okay, more acidic means a lower pH. Okay, so in tissue, you're going to have a lower pH when that tissue is active, right? So you can think of like our lactic acid production, right? You can associate those two things in your mind, kind of. Okay. So when we see a decreased pH, what's going to happen is that we're going to see a shift to the right in this saturation curve. So when we say a shift to the right, we mean we're going to be doing something like this. Okay. So a shift to the right is going to mean that we are going to more easily unload oxygen to the tissue. So a way I like to remember all these effects actually is thinking about tissue that's active. I find it very helpful to think about, okay, when this is going on, would you want to unload oxygen to tissue, or would you want to like keep your oxygen in your blood? Most of our shifts that we're going to be talking about are going to be rightward shifts. So you can see here this example rightward shift I drew. So now the veins and systemic arteries would still be at their same partial pressures of oxygen. You're still breathing, right? So we haven't changed that part with the pH, right? So we're still going to have our partial pressure of 40 and our partial pressure of 100, okay? But now, if we have a rightward shift, move this curve to the right, at a partial pressure of 40, which is what we have in the veins, right? This example curve I drew, we only have 20% saturation of hemoglobin, okay? Systemic arteries still have high saturation because of that higher partial pressure, depending exactly how we draw this curve. So when we're talking about that unloading, before we were comparing just this 98.5 and 75% saturation, right? That was our original hemoglobin unloading. Like that's how much we let go of. But now we've let go of a lot more. So we're still just going to compare how much was in our systemic arteries. What was that saturation? To what's in our veins. But you can see this is an awful lot more oxygen that we let go of when we compare those 2 points. So that's how this idea of unloading is related to that curve. Okay. So if we unload more oxygen to the tissues, what we're saying is we see a greater difference between these two numbers. Okay. So there are other factors that have similar effects. So one is the effect of carbon dioxide. So we call this the carb amino effect. So carb amino carbon dioxide is how I would remember that. Okay. So what happens here is that carbon dioxide does react with hemoglobin. Okay. When it does that, we get something called carbamino hemoglobin. Now this has a lower affinity for oxygen than hemoglobin just normally does, than the deoxyhemoglobin would. Okay. So lower affinity means it doesn't want to bind oxygen so much, right? So this means that we are going to let go of oxygen more easily than we otherwise would, right? They're just like weaker magnets for each other when they have this lower affinity, right? So this means that likewise, when we see increases in carbon dioxide, okay, we see an increase in carbon dioxide. This is also going to follow the law of mass action. So we're going to push this equation to the right. More carbon dioxide, we form more carbamino hemoglobin. Carbamino hemoglobin doesn't like to bind oxygen as easily, which means that when it gets to the tissue, it also lets go of that oxygen more easily. Okay, so we unload more oxygen in the active tissue. And something that's kind of amplifying this effect, right, causing this effect, is that when you have increases in metabolic activity, we're really talking about making ATP. So we're talking about breaking down glucose. Carbon dioxide was our byproduct of that. Okay. So we see increases in carbon dioxide in tissue that's actively making ATP. So this effect is going to take place at that tissue, which is great because if you're making lots of carbon dioxide, this waste product means you're making lots of ATP. You're probably using a lot of oxygen. You want to get that oxygen more easy. You want it to unload more easily. So we'd also see a rightward shift, just like that last one here. So a right shift here means that we're unloading more easily. Okay. This next one has a long name, but we do have an effect of a molecule called 2,3-diphosphoglycerate or 2,3-BPG. And 2,3-BPG, usually when we talk about it, we're thinking about anemia, or very, very often we're talking about high altitude. So there are actually adaptations to how much of this you make, depending on what environment you're in and how long you've been there. Okay. But basically, this is a molecule that red blood cells make when you have a low oxygen environment. Okay. So they make a bunch of this. Okay. And what this does See, your body's in this low oxygen environment. You don't have enough oxygen kind of in the air you're breathing, right? But you still want to get oxygen to tissue. Okay. So what happens here is that when you have this molecule, you're going to decrease the affinity of hemoglobin for oxygen. Okay. So that even though there's less oxygen around, you can still unload a bunch of oxygen to go to the tissue. Okay. So this is useful if you're anemic or if you're at a really high altitude. You don't want to get altitude sickness, right? This is part of why if you're going somewhere with really high altitude, they tell you to sort of stick around at the bottom of a mountain for a day or so while you adapt, okay? So this is so that you can produce this molecule, so that you can unload oxygen more easily, so that you can keep getting oxygen to your tissue, even if you're not breathing that much in. So this also is a rightward shift, okay? Now, one time that we talk about leftward shifts is when we're talking about carbon monoxide. So one of the dangerous things, there are a couple dangerous things about carbon monoxide, but the overall problem with carbon monoxide is that it's going to prevent you from carrying enough oxygen in your blood. Okay. So one of the things that's happening there is that hemoglobin is really getting full of carbon monoxide. It likes it a bit better than oxygen. Okay, so we have carbon monoxide actually kind of binding in to those sites where oxygen would be. So this means that you're not going to see that higher saturation of oxygen, even if there's oxygen around you, because it's being inhibited by this carbon monoxide. It's binding to that instead. So this is going to prevent oxygen from binding to hemoglobin. So that would go the other way. Okay. A note, we do talk about all three of these effects. Usually we're talking about a rightward shift when we're focused on the tissues. But if you stare at these long enough, you may also realize that if you do the opposite, you can have a leftward shift. So, right, if we go back to this Bohr one, right? So we're talking about if we have pH going down and producing a lot of acid, we had a rightward shift, right? If the opposite change happens and you're for some reason having a more basic environment in your blood, right, we'd see a leftward shift the opposite direction. So these can go both ways. We focus a lot on the rightward shifts specifically because they happen at metabolically active tissue, so they're important to your everyday functioning, but they can go both ways. Okay. Cool. All right. So we're going to move on and talk about carbon dioxide next. Okay. So carbon dioxide is largely going to be carried as using a molecule called carbonic anhydrase. It's a different situation than we have with hemoglobin and oxygen, because we're not like binding two things together and carrying them around like in a car. We're really going to be like converting carbon dioxide using this enzyme, and then those products are going to be circulating in your bloodstream. Okay, so carbon dioxide is going to get exchanged at the lungs and at the tissue, just like oxygen did. Okay, so you could trace through our partial pressures for carbon dioxide, just like I did at the beginning for oxygen. Okay, but fundamentally, what's happening is we breathe out carbon dioxide at the lungs. So we're going to have lower values of carbon dioxide in our arteries until we get to our tissue, where they're going to take in carbon dioxide that's being created in that process of making ATP. And then we're going to have that higher partial pressure of carbon dioxide as we return to the lungs before we breathe it out. Cool. So when we're transporting carbon dioxide in the blood, okay, we actually have more carbon dioxide dissolved just directly in the plasma as carbon dioxide than oxygen. So oxygen is really in the hemoglobin, but carbon dioxide is partially dissolved in the blood. Okay. This is breaking down for us kind of where some of our carbon dioxide is. Okay. So we had 1.5% was the relevant comparison to the oxygen. That's not on this chart. What this is showing us is that carbon dioxide is partially in the blood as carbon dioxide. Part of it is converted to bicarbonate. Okay. So that's a lot of what's going on in our blood. So we've converted carbon dioxide essentially. And a little bit of it is bound to hemoglobin. So it doesn't bind at the heme group at that same site as oxygen, but it can bind into hemoglobin, just at other parts of it. That's part of why we could have that carbamino effect. Right. So the carbonic anhydrase is not like directly binding and carrying the carbon dioxide. What it's doing is converting it. Okay. So carbonic anhydrase, it's an enzyme that catalyzes the reaction of carbon dioxide and water into carbonic acid. And then carbonic acid, I'm just taking this first one step further, dissociates when we're in this aqueous environment to hydrogen ions and bicarbonate ions. Okay. So when we saw on that previous chart that we're carrying a bunch of carbon dioxide as bicarbonate, okay, this is how that happened. So we took our carbon dioxide, mixed it with water, carbonic anhydrase turned it to carbonic acid. And then because we're in a liquid environment, an aqueous environment, that dissociated into our two ions here. Okay. So overall, if we have an increase in carbon dioxide, we're going to increase our carbonic acid. And if we increase our carbonic acid, more of that is going to get converted to our hydrogen ions or protons and our bicarbonate ions. So more carbon dioxide means that this process is moving to the right. So there is a relationship between the partial pressures of oxygen and carbon dioxide. So we looked at it from the other direction when we were talking about the carbamino effect. But here we see kind of the effect of oxygen on carbon dioxide. So what we're looking at is our partial pressure of carbon dioxide, okay? And how much carbon dioxide is total in the blood. Okay? So if we picked just this center curve, right, you'd see that at a partial pressure of carbon dioxide, any given partial pressure, you have a corresponding total amount of carbon dioxide in the blood. That's why we have a curve like this. Okay? So we've picked out one point on this normal central curve. Okay, so this represents what's happening in our veins when the partial pressure of oxygen is 40. Okay, so this is how much total blood carbon dioxide we would carry here. I'll make that at the same level. Okay, but we're going to carry different amounts of carbon dioxide at different partial pressures of oxygen. So we can see that when our partial pressure of oxygen is 100, like it is in systemic arteries, the blood isn't going to hold the same amount of carbon dioxide. Right? So that's why this curve is lower here. Okay? And then if we were to have a very low partial pressure of oxygen, we'd have a lot more carbon dioxide. So these two things both affect each other. Carbon dioxide affects oxygen, oxygen affects carbon dioxide. So when we're looking at the blood overall, and when we're thinking about transport of these gases, what we're really trying to figure out is how easy is it for us to get oxygen onto our hemoglobin in the lungs? How easy is it for us to unload that oxygen at tissue? Okay. And we're also looking at how much carbon dioxide are we carrying? Are we getting enough of it to the lungs so we can breathe it out? Okay. So if we're looking at the blood, we might trace either our carbon dioxide or our oxygen or all two together. So this is showing us overall everything that affects oxygen unloading. So this whole graph is showing you effects that increase unloading of oxygen. So basically increase how much oxygen gets into your tissue. So all of this is showing us stuff that's going to ultimately increase how much oxygen is available for making ATP. So we see some direct effects of oxygen. We see effects of carbon dioxide. We see that carbamino effect, and we see the Bohr effect here. Temperature would also affect this potentially. Actually, temperature does affect this, but overall, that's kind of what we're looking at here. I wouldn't worry too much about the total diagram as it stands. Here we see some opposite reactions. So these are reactions we would have. All this changes the arrows basically, right? So before we were seeing reactions to drops in O2, now we're seeing reactions to the opposite, to rises in O2 and drops in carbon dioxide. Okay. So that's what we have for today. We'll do a little bit of practice about this after break, but we're going to be moving forward into thinking about our renal system and our kidneys pretty immediately. So if you didn't watch that video on Wednesday going through the anatomy of the kidney and specifically the anatomy of the nephron, make sure that you do that at some point before I see you next, because I want you to know what I'm talking about when I start referencing all sorts of different tubules and stuff. Okay. So that's where we will leave it.